Hi everyone. All right, in the previous video we talked about P53 and its interaction with apoptosis. In this video I'm going to cover some therapeutics that can be used kind of to maximize this apoptosis process. So our first one is going to be by activating death receptors. Right, so we are, in this case, going to, let me just draw out kind of my image for this. This is my cell membrane. Right, and I'm going to have a TNF receptor. And we can have therapeutics that are going to come in and activate this TNF receptor, but they're not necessarily the endogenous TNF molecule. So an example of this is dulanamine, which is a trail ligand analog that mimics that TNF molecule and can bind to the TNF receptor, leading to this downstream apoptotic signaling pathway. So this is kind of increasing extrinsic apoptosis signaling is the strategy for this. A second method can be increasing transcription of pro-apoptotic factors. Right? So when we go to transcribe our pro-apoptotic factors, maybe BAX is a good example of that. One thing that we need to be able to do is actually physically access the gene, and part of that is having the gene be acetylated. We won't go into a lot of detail, but essentially to think about it, when our DNA is acetylated, it's stretched out and easily accessible. When it's deacetylated, it kind of ends up being wrapped up in these coils that makes it really difficult for the transcriptional machinery to access it. So we can actually inhibit this deacetylation process with therapeutics. An example of this is SAHA, which is a histone deacetylase inhibitor, which, just like it sounds, it prevents this deacetylation kind of transition. So maybe a more accurate way to represent this is that normally we need to transition from acetylated to deacetylated. I move this up. SAHA is going to inhibit that transition process. And this is particularly going to be effective for increasing are pro-apoptotic proteins. So it's going to be a somewhat specific process. So that's our second kind of way that we can increase apoptosis. We can also inhibit apoptosis inhibitors, right? So we talked about IAPs, which are essentially, if we just draw them out, IAPs, they're inhibiting the transition of procaspase 9 to caspase 9 to its active form. That's the process that we're inhibiting with IAPs. We can take these molecules called polyphenol ureas, which essentially mimic smack diablo. And these are going to inhibit IAPs. So that's kind of our example of how we can decrease our apoptosis inhibitors, these polyphenol ureas. And then lastly, and kind of one of the more interesting therapeutics is BH3 mimetics. And a big example of a BH3 mimetic is going to be venetoclax. And this is going to mimic our death helpers. Venetoclax. So let's walk through what venetoclax is capable of doing. The main activity that venetoclax has is it inhibits BCL2. Now, important to note, this is one of our pro-life proteins. Venetoclax is specific to BCL2. So this is specific. So it cannot interact with BCL-XL or MCL2 or other of our pro-life proteins. It's only going to inhibit BCL2, and kind of downstream of this, it can increase the activity of our death helpers in both the kiss and run activity and also in inhibiting BCL-XL, because venetoclax is kind of taking care of BCL2. That's our big example of our BH3 mimetic that is going to be inhibitory of kind of those pro-life proteins. So those are our four therapeutic techniques that we can use. We can artificially activate our death receptors. We can alter kind of the transcription of our pro-apoptotic factors. We can inhibit our apoptosis inhibitors. In this case, we're mimicking smack diablo with these polyphenol ureas. Or we can mimic our death helpers and inhibit those pro-life proteins. All right, and those are my kind of videos on apoptosis. I hope you enjoyed watching.